For Professor Pratt's microbiology class, Cass and I were given an unknown bacteria. We performed several metabolic tests in order to determine our unknown substance given the dichotomous key we made previously in class. Our first test was a gram stain. During this test, we found that our unknown substance was pink, meaning it is gram-negative and bacillus in shape. This narrowed our search from 26 down to 13. The next test we performed was a lactose phenol red test. This tests for fermentation of lactose. This also tests whether or not a gas was produced. The test is differential for oxidation or fermentation. If lactose is fermented, the liquid will turn from red to yellow, and if a gas was produced, you will see a bubble within the tube. We found that our unknown did ferment lactose and also produced a gas. Since our unknown was gram-negative, bacillus, and a lactose fermenter, we were able to narrow our search down to 4 types of bacteria. These bacteria were C. fruindiae, E. coli, K. pneumoniae, and E. coliaceae. A sucrose phenol red test is similar to the lactose phenol red test, but the medium is sucrose and tests for the fermentation of sucrose. Just like the lactose phenol red test, a positive result will turn from red to yellow, and if a gas was produced, there will be a bubble within the tube. We found that our unknown did not ferment sucrose and no bubble was formed. After performing the sucrose phenol red test, we were fairly certain which bacteria we had. After we narrowed our results down to the 4 options, the sucrose phenol red test left us with only 1 bacteria matching the results we had obtained. We concluded that our unknown bacteria was E. coli. We then performed several tests in order to verify our results.
A glucose phenol red tests for the fermentation of glucose. The medium is glucose. This test is differential for determining oxidation or fermentation. It also tests for whether or not a gas was produced. If glucose is fermented, the liquid will turn from red to yellow, meaning an acid was produced. If gas was produced, there will be a bubble within the smaller tube. We found that our unknown did ferment glucose and a gas was also produced. This means that our unknown substance is a heterofermenter of glucose. A triple sugar iron test indicates the presence of glucose in either sucrose or lactose fermentation. If glucose was fermented, the bottom half of the agar would turn yellow. If either lactose or sucrose were fermented, then the top half of the agar, or the slant, would turn yellow because enough acid was fermented to turn the whole thing yellow. The ferrous sulfate helped us to determine whether or not the fermentation produced gas. In our case, the entire agar was yellow, meaning that the unknown produced glucose and either lactose or sucrose. This backed up our previous evidence of our unknown being positive for the glucose and lactose results. Also, gas bubbles were formed in the TSI agar, meaning that there was gas present. This also positively correlates with our previous tests, glucose and lactose being positive for gas. The test for oxidase is differential and tests for the presence of cytochrome oxidase. A positive result is purple and means that the bacteria uses oxidase in the electron transport chain. A negative result is yellow and means the bacteria uses oxygen. Our unknown was negative for oxidase, meaning it can use oxygen for the final electron acceptor. The test for catalase is differential and tests for the presence of the catalase enzyme. In order to do this, we placed a small amount of our unknown onto a slide and added hydrogen peroxide to it. If bubbles were formed, then the test was positive. Since we were pretty sure that our unknown was E. coli, we knew that the test should have been positive, and it was. This reassured us that we were correct. The thioglycolate test was a test for the type of growth of our unknown. It determines the oxygen requirements of a bacterium. The broth contains sodium thioglycolate, thioglycolic acid, L-cysteine, methylene blue, and 0.05% agar. Since our results showed growth at the top of the test tube and throughout the test tube, just not at the bottom, it could be said that oxygen was not required, but it did not stop growth from occurring when present. Therefore, we said that our unknown was a facultative anaerobe, and E. coli is indeed a facultative anaerobe.
The test for Simmons citrate is differential and contains a pH indicator. It tests for the ability of bacteria to use citrate as its sole carbon source. A positive result is blue, while a negative result is green. The change in color indicates a drop in pH. Our unknown was negative for citrate. The test for gelatinase is differential for proteases. Proteases are proteins that are used as a source of growth. A positive result transforms the nutrient broth into a gel. A negative result will stay in a liquid form. Our unknown was negative for gelatinase.
Indole is differential for the production of indole, pyruvate, and ammonia. To perform this test, we put our unknown into tryptone broth and added the indole indicator. If a red or pink band was produced at the top of the tube, the unknown is positive for indole. The absence of a band means the unknown is negative for indole. Our unknown tested positive for indole. After performing all of these metabolic tests, we were able to conclude that our unknown was E. coli. We then performed a Kirby-Bauer assay with 7 different antimicrobials and chose the best disinfectant and antiseptic from the results. Old 10% bleach, or number 2 on the plate, is successful in removing E. coli from surfaces. It is a disinfectant because it should not be used on the skin and can be harmful. The zone of inhibition was 18 millimeters on the plate, meaning that it did not allow for growth up to 18 millimeters. Iodine, or number 3 on this plate, is successful in removing E. coli. It is an antiseptic because it can be used on the skin and often is during surgery. The zone of inhibition was 20 millimeters on the plate, meaning that it did not allow for growth up to 20 millimeters.
Next, we also performed a Kirby-Bauer assay with 7 different antibiotics. I will explain the results of 3 different antibiotics. Number 7 in the center shows erythromycin. E. coli is resistant to erythromycin and has a 0 millimeter zone of inhibition. Erythromycin falls into the category of a macrolide antibiotic. Macrolides bind to the 50S subunit of the ribosome and inhibit protein synthesis. Macrolides are bacteriostatic and do not kill bacteria, but only inhibit growth by targeting gram-positive cells. Number 3 on the plate shows penicillin with a 0 millimeter zone of inhibition. E. coli is resistant to penicillin. E. coli is gram-negative and it has a thin peptidoglycan layer within the wall. Penicillin is a beta-lactam antibiotic which targets gram-positive bacterial cell walls in order to break down the bacteria, and it works by inhibiting cell wall enzymes needed to synthesize peptidoglycan. This means penicillin is unable to kill gram-negative E. coli. Lastly, number 1 shows E. coli and its sensitivity to ciprofloxacin. The zone of inhibition was 40 millimeters, meaning it was able to kill E. coli up to that point. Ciprofloxacin falls into the category of a quinolone antibiotic. At the molecular level, the ciprofloxacin interferes in the enzymatic process of gyrase that causes DNA to unwind. This stops the synthesis of DNA and protein synthesis. As a result, the growth of bacterial colonies is stopped. I would use ciprofloxacin to treat an E. coli infection. We concluded our unknown bacteria as E. coli through several metabolic tests. We found that ciprofloxacin will be the best antibiotic to treat an E. coli infection. Old bleach is a good disinfectant against E. coli, and iodine would be a good antiseptic.